recording begins. It is July 14th. It's a Monday. It's 2025. Cool. It's good to see you all again. Hope the 3-day weekend was restful. Let's go ahead and do our regular thing where we look at the course schedule and then this week's module. And then we'll just get into lecturing. We've got a cool lab today where we're going to be extracting proteins from these E. coli. And you're going to be looking at protein concentrations using something called a colorimetric assay, where you react a colorless substance like protein with another chemical that makes a product that has color. And then we look for the amount of that product using what technique? Spectrophotometry. Points, 4 points, 4 John points. So this time, though, you don't have to use the Spec 20s or the Spectromax 200s, because we're going to go upstairs, get to check out the core research facility, and use the 96-well plate reader, which is essentially just a spectrophotometer that can work way faster and costs way more money. So that'll be cool. All right? Course schedule, here we are. We are here in week number 2, talking cell structure and function, 2 assigned readings, 1 assigned reading quiz, but Chapter 7 material will be covered next week in a Chapter 7 and 8 reading quiz. Protein extraction lab today, enzyme reaction rates lab tomorrow, carbohydrate assay the day after that. This carbohydrate assay will also be a colorimetric assay, but we'll be using the Spectromaxes for that, okay? That's that. Week 3 is when we have our first midterm, well, our only midterm on Thursday for lecture material, and our only, our lab midterm, effectively, okay? And that's going to be covering material through the end of week 3, through the Tuesday of week 3, all right? So next Thursday, basically just come in and do a lecture exam. Yes, question, please. Not for the lecture exam, unless you have accommodations through the SDRC, or it's the CDRC now, I think is what it's called.
Yes, the student part still called the SDRC.
Okay, cool. Yeah, the acronym, I get mixed up, you know. If you have accommodations, though, for a test sheet, like a note card or something, then you can reach out to them to get confirmation before next Thursday, and I will, get that email and totally allow it. Question?
Usually they leave emails for that kind of stuff at the beginning of the semester, because everything's a bit different here. Should we manually contact them?
Yes. For the summer, everything, just to repeat that, normally I receive emails saying, these students have accommodations for you. In the summer, it doesn't really go like that. So it's best if you reach out to them directly. Yes. And if they're really giving you a hard time or they're not getting back to you, then just talk to me directly, okay? I believe you. I don't think anybody's trying to get one over, all right? Yes, please. The format of the exam, great question. The lecture midterm is all multiple choice. It will be on Scantron forms. Some of you maybe haven't used Scantron forms before. They're less and less popular. You're in that category? Yes, please. I'm so happy this is all coming up right now. I stockpile them, so I've got some backups, but ideally it would be great if you could get your own Scantron 882E forms or equivalent, okay? You need 2 of them. One for the midterm, one for the final. And I'll have enough to probably get us through the midterm, but if nobody has one on the midterm, I'll be out by the time the final comes, Yeah, sometimes they can be a little hard to track down. Yes?
Do you know if both bookstores are open, or which?
I'm not sure if the bookstore should be open, right?
One on campus here is.
Okay.
Awesome.
Okay.
So go on lunch.
Go on lunch or go on Thursday.
Yeah.
Yeah. So the on-campus bookstore, just to reiterate for anybody who's watching the recording, the on-campus bookstore is open until 3 p.m. They should have those Scantron forms. During the regular semester, you can get Scantron forms at the testing center. They'll usually be kind enough to give you a couple. Also, Rose House will often have a stockpile of Scantron forms. And Rose House is a resource for additional supplies if you need them and can't afford them. All right? Question? Okay, I'm hearing that the off-campus one may be open until 5 p.m. That's the one off the plaza, right? Okay, and they should have these Scantron 882E forms. But if you can't find one, I'll have backups for the midterms. Yeah, you can also order them online. If you buy like a pack of 15 or 20, that's the best way of going about it cost-wise. But I don't know if you want to coordinate that with everybody else in the class. Though if you ever take another class from me, I love Scantron multiple choice exams because I can turn them around, get them graded real fast. I can't put my own bias into that machine, the robot that grades the exams. And I don't know, personally, I like multiple choice exams, but that's just me. Yes. Now the format of your lab midterm, there will be a couple of multiple choice questions, maybe a true or false on there, but mostly it's going to be open space where you have to carry out a calculation or interpret some results. Sometimes your answer might just be a number, but you can write as much as you want in the open space, and if you get close, like you're doing every step right, and then you forget to convert correctly from milligrams to micrograms, I'm going to give you partial credit. So the idea is in the lab, there's opportunities for partial credit. Cool? This is good stuff to get discussed. If I was doing my job perfectly, but whoever does their job perfectly, I would have discussed all of this with you on the first day of lecture. But back then, who knows if any of us are ready to memorize any stuff. So, does that feel good for, remember, next week, the midterms? Any other questions? Yes, please.
Will there be any formulas provided
On the lab?
exam?
The lecture exam won't require any math beyond a question about dehydration synthesis and hydrolysis, which is, basic, just subtract, There will be math on the lab midterm, but it's open notebook. So all the equations can be found in your protocols. I have basic calculators that will be available for you. They're just very straightforward, like little blue teaching calculators. But if you want to bring your own calculator, that's great. I don't usually allow people to use phones as calculators for obvious reasons, Was there another question here?
It was about the type of calculator. Do we have to use like the basic one, or if we have a graphing calculator?
Bring a graphing calculator if you want. There'll be a standard curve question on there, and if you prefer to do standard curve questions in your graphing calculator, where then you can enter in a value for y and solve for x like that, you totally can. Or if you want to just rearrange the equation on your piece of paper and then do it with the basic calculator, that's a viable option too. Awesome. Any other questions? Cool. Even with a little late start and some extra, questions and clarifications, we are doing great. Normally at this point in the summer, I'm like 20 slides behind, and we're right on where we need to be. So we can just continue at that pace, and we'll be just fine. Here's this week's module. You know how the modules work now, right? Here's the PowerPoint. I'm going to download it. Make sure that we all have access to the same PowerPoint. One reading quiz for this week, just the one. There'll be 2 next week. Here are the lab materials. In these labs that are a little more evolved, you'll see there's actually like a lab page. And I'll have posted tutorials and tutorial pre-lab lecture sort of materials. These are from quarantine times, And here's a tutorial of how to use the SpectraMax machine that we'll be using upstairs. I just have additional video resources usually in these pages, okay? Protocols, don't forget, print protocols, begin the lab, 1, 2, 3 this week. Lab assignments, 1, 2, 3, one for each lab, right? Questions?
Last week you had said you were probably gonna update the labs. Are they all ready to print?
Just go ahead and print them all, you know. That's kind of a pipe dream that I'm going to actually sit down and edit and update all the protocols when we're here in person and I can just address with all of you like, oh, this says to do this step. Let's actually run it this way. Yeah, so you're good. The protocols, I don't expect them to change. It's also what the lab prep people have. It takes a lot of coordination to get all of the right stuff in here. And if I make a last second decision like, we're actually not going to use the current extraction protocol for the proteins, we're going to mix it up, that takes communication between multiple levels of people and sources that I ordered previously. So I don't, you know, the short answer to your question is yes. The answer to your question that makes me look better is, oh yes, I've already updated them. They're all ready for you now. Go ahead. Let's go with that one. Okay? Yeah, I spent all weekend doing that. Thank you. You shouldn't, but that's okay. Here's the reading recordings. It's awesome if you can listen to these. There's a lot of cool stuff in chemical biology. Honestly, it's a good textbook. Just do it during your commute or a hike. Listen to my voice. There's also this is a pedagogy trick. I like to give you pedagogy tricks. The more a group of students hears your voice, the more they're likely to trust what you say, but like, I mean in a way of internalize what you say, like take the information in. It's one of the reasons that they say having the same lecture and lab instructor is so important. It's interesting stuff, but all that sort of research on humans are so complicated. You never, you know, you're really just going to trust it like right off the bat? I don't know. Okay, y'all excited to start talking about cells? Oh, yeah, we are. Oh, we're pumped. Check it. This week's only 80 slides. So that means we'll spend a little more time on the individual slides. And if we get too far ahead, who knows? Maybe Thursday's a short lecture, or maybe Thursday's not happening. But I don't want to make any promises. You're going to get your full money's worth, though, I promise, because this has all the material you would get in a normal 15-week semester. Yes, enable external content, because there's little video things. Okay, ready, folks? All right. So we started zoomed in all the way down to the level of the atom. We talked about the basic subatomic particles. Then we talked about the types of forces that hold atoms and molecules together. Then we talked about the macromolecules, right? The big molecules that are important for biological function. We talked carbs, we talked proteins, we talked lipids, we talked nucleic acids, right? We did all of that in 1 week. Now we're continuing to zoom out, okay, and looking at cells, okay? Now, cells, like anything, are built up of these molecules, and so all these biological macromolecules we talked about are going to come together to form the primary unit of life, the cell. Learning objective slide, there it is, okay? First, brief discussion of how do we actually study cells, right? How do you actually look at them? A lot of science, especially chemistry, can be done without directly observing anything, right? You can't actually really see most molecules in any way, so you have to infer what's going on through sort of indirect evidence, right? And a lot of assumptions. But with cells, well, cells can be visualized. We can see them doing things, at least things like changing shape or growing. We can even see the movement of subcellular organelles, all right, or at least the arrangement. Anyway, I'll get into the key distinction between being able to see the movement and the arrangement. So here, light microscopes. We used them last week, all right? Light microscopes use light. We see things because our eyes can detect light, all right? Light is radiation. Cool? Solid stuff here. So light's radiation, our eyes are adapted to seeing different wavelengths of light, interpreting them as colors, all right? Light microscopes have a limit of how much magnification you can achieve based purely on how light actually behaves. So remember when I described to you the electron orbiting the nucleus? And I was like, it can only exist at certain orbitals because it's really a wave. And at certain distances, the wave reinforces itself because peaks coincide with peaks, all right? If it's any closer, the wave interferes with itself, all right? A lot of small particles behave like, a lot of small things behave like particles and waves at the same time because both particles and waves are models to help us understand things. We can understand a wave because we can see it. We can understand a particle, we understand the concept of a little discrete unit of something, right? But really little things, all right, don't behave like either, okay? And you're familiar as a college student, somebody has shown you the double slit experiment, talking about light and how the light can go through, you have to slit 2 slits of paper, the light only goes through 1, and it's like, why did it do that? You know, oh, because it's interfering, it's a particle wave. Maybe you've heard something about that, right? One of the things that light does is different light particle waves traveling next to each other interfere with each other, all right? So that makes things blurry. When you zoom in far enough with a light microscope, the actual light itself prevents you from seeing things any more clearly. So you have a limit of what we would say resolution and a limit of magnification. So light microscopy, because of how light is, not because of the equipment being out of date or anything, has a limit of about 400 times magnification to be clear, and about 200 nanometers resolution. And what resolution means, all right, which you've heard of, say, a high-resolution monitor or television, something like that. Resolution, or a high-resolution image collected with a camera, right? Resolution has to do with how close 2 points can be together, and you can still tell that they are separate points. So this means that with light microscopy, 2 points can be 200 nanometers apart, and you can tell that they are separate points. Does that sort of make sense? If they're any closer, they look like 1 point. Now, if light's the problem here, we need a way to look at small structures without involving light. And that's where we use electron microscopy. We have, I think at this point, 3 or 4 electron microscopes on this campus. There are a couple of different types of electron microscopy, and I'll tell you about them. But if you look right here, it's a much more involved piece of equipment. They look different from this now, of course. They look a little fancier. They look more like minimalistic, you know, because that's what translates to scientific now, is less dials and meters and clicks and beeps and more smooth, chromed up plastic, you know. But it does the same thing. You take a sample, you pop it in this little sealed chamber where no light can enter, because light is a problem here. Your sample gets blasted with electrons, okay? And then those electrons, imagine that you're shooting the electrons at something, and behind that thing is a little sensor. That sensor detects how the electrons get scattered when they pass through the sample, okay? So a computer is then needed to take the scattered electron patterns and convert that into an actual image that you can look at through the eyepieces here, or that you can collect and develop onto a photograph. So in electron microscopy, light never enters the chamber where the sample's being viewed. This gives us way higher magnification and higher resolution. This isn't 50 nanometers, this is 50 picometers. And there are 1,000 picos in a nano, all right? Awesome. So any questions overall about that? If you think microscopy sounds interesting, if you liked looking through microscopes, and if you would like to look through more expensive microscopes, we have courses in electron microscopy. You want to start trying to get in early because they fill up fast, and they give higher standing to people. And if you're later in your degree, but sometimes you can get in there. And being an electron microscope technician can be a whole career, where you just collect images, really good images, for people who are trying to look at subcellular processes. All right? It's cool stuff. And you can get trained how to use the modern technology here at Cal Poly Humboldt. We just got a new one because of a big grant, and it's very exciting. All right. Light microscopy versus electron microscopy of the same thing. All right, so look here in light microscopy, we've got salmonella. Looks like these little purple dots. This is max magnification. So you could say, oh, I think there's salmonella in this sample. There's little purple dots there. Now this sample is actually this color. It has been stained. It has gone through a staining process to make the salmonella pop out in the sample and look purple. If it hadn't been stained with dyes, it would look like clear but bleary. You know what I mean? Now, earlier when I was like, color, we only perceive color because we're looking at different wavelengths of light. I was trying to lay groundwork for this slide here, where you look at this image on the right. This is a black and white image. There is no color when you're doing electron microscopy because there's no light, all right? You're just looking at scattered patterns of electrons that bombarded a sample. So if you see electron microscopy image with color, it has been colorized. Just somebody photoshopped it to give it color, to give a better idea, to give contrast, so you can see what was going on here. All right? It's a good thing to be aware of with electron microscopy. The images, due to the technology and the technique, cannot have natural color because no light is introduced into the sample. Cool? But look how much closer you can get here. You can get a real good look at these things, the tissue that they're embedded in. You can see these little, I forget what these are called, the lazy ways to make these little fibrils, but that's lazy. But these little connective things, they're trying to form up, they're trying to group up and make a little blob of themselves so they can become pathogenic. All right. What can we look at with different techniques? Well, with the unaided eye, you can see some cells. You can see a human egg cell with your unaided eye. You're not going to be able to see it very clearly, but you could see it if it was in a petri dish and someone was like, you have to do this, because that zooms in by like 4 times just doing that, right? And you look at the petri dish, you can see it, right? But you can't get down into any of the subcellular structures until you're using light microscopy. Now, some nerve and muscle cells can be real long. Like you can extract them and then wrap them around a toothpick and like see them pretty clearly, just chomp it down. You know what I mean? Because some nerve cells have to, say, run from the top of your head to your toe, all right? And there are big whales in the ocean, and some nerve cells have to run all the way from their head to the tip of their tail. So that's a big cell, and if you can extract it out, yeah, you're going to be able to see that with the unaided eye pretty easily, right? So, human. You can see a human with the unaided eye. Then you got to get down into light microscopy. There's some overlap here saying that, yes, well, you could see a frog egg or a human egg with your naked eye. You can't see as much detail without microscopy. You want the detail. Light microscopy can get you pretty far down. You're going to be able to see bacteria, okay? Most bacteria down to the smallest bacteria you might not be able to see very clearly with light microscopy, but there are techniques for super resolution microscopy where you either have a computer process the image additionally to remove the blurriness, or you control how the light is blasted onto the sample, and that gives you different, higher resolution. So with more expensive light microscopes, you can get down to see smaller things. But if you want to see the very smallest things, like viruses, ribosomes, proteins, lipids, right, you need electron microscopy. Small molecules, you can't really see a molecule of water, all right? But you can see a phospholipid with electron microscopy. Anyway, domains of, that's the microscopy piece, okay? There's a ton of techniques here. Normally in the fall semester, I have a slide that has like 20 images collected with different types of microscopy, but we're just not going to worry as much about that. We're going to focus on the difference between light microscopy, electron microscopy. Just know that there's more variety out there, okay? There's more variety out there. There are very expensive microscopes that can get closer, that can get smaller, higher resolution. Cool? So you don't have to use an electron microscope. But there'll always be a need for electron microscopy because you get a more direct view of things. You don't have to use as many. Anyways, I'm getting off on a tangent. Electron microscopy. If you like microscopy, it's a career track. You could do it. You could become the best electron microscope image collector, and you would be sought after. And you could travel around and go to different universities that were doing research and be like, well, you're using this electron microscope. I'm familiar with it. Let me just tweak these settings and look at the image I've collected for you. And they go, ooh, and then they publish with your results. It's pretty cool. All right. Domains of life. We've already briefly talked about eukaryotes, prokaryotes, eukaryotic versus prokaryotic cells. Now we're going to get into more depth with them. You know how I feel about repetition. I'm doing it right now. Prokaryotes include the domain bacteria and the domain archaea. I'll never live down when I called them kingdoms. Eukaryotes are in the domain eukarya. Okay? So, bacteria, archaeans. Eukaryotes, all the big multicellular complex things. Part of why they can be so complex is because they have nuclei, right? They can have a lot more control over what genes are getting expressed, all right? And they have these endomembrane systems, all these subcellular membrane-bound organelles, which means they can have specialized compartments of the cell to carry out functions at way higher efficiency. It lets them create more energy and have more order, and that single cell level of enhanced order allows for more multicellular enhanced order. Cool? Prokes and eukes. Prokaryotic cells. Remember, karyos is Greek for kernel. The karyo, the kernel that we're talking about, is the nucleus. Prokaryotic cells, you could think of them as having like a prototype nucleus, right? Like a pro-nucleus, right? Because they do still condense their genetic material into a certain region called the nucleoid, okay? But the nucleoid is not membrane bound. Nucleus is membrane bound. In prokaryotic cells, there are no membrane bound organelles. However, there are ribosomes. You need ribosomes to generate proteins, if you remember that from our previous lectures. Central dogma, right? DNA, RNA, protein. The last step of RNA to protein is facilitated by ribosomes, which are organelles in that they are sort of multimolecular complexes, large multimolecular complexes that carry out a very specific function, have specific parts to them, right? That's what we're talking about with an organelle. When you're thinking about how do I remember what an organelle is, how do I define an organelle? Just think of it as a little organ, you know? Like, you know what your organs do. They have specific functions, they have specific parts to them. Organelles are no different. They're just little. All right? Cool. Cytoplasm. All right? Cytoplasm means like cell fluid, cell juice. So the cytoplasm is all the liquid inside of the plasma membrane. Now, when you have membrane-bound organelles, this I don't want to get too confusing with it. I hope I can avoid it. Here, everything is floating in the cytoplasm. The DNA, the ribosomes, everything. It's just floating there in the fluid, the intracellular fluid in the cytoplasm. But in a eukaryotic cell, where there are membrane-bound organelles inside, right? You've got a membrane-bound organelle, which has fluid inside of it, and outside of it is the cytoplasm. Or outside of the nucleus is the cytoplasm, but then inside of the nucleus is its own fluid. It's no longer it's not the cytoplasm. The cytoplasm is just the fluid inside of the plasma membrane or the cell membrane, all right, that all the things are floating in. Does that kind of make sense? Question, please.
The nucleotide doesn't have its own separate fluid?
It's the nucleosome. Yeah, the nucleoid, you mean.
Yeah.
Yes, the nucleoid, it does not have its own separate fluid. It is floating directly there. And that will be incredibly important when we start to talk about how transcription and translation occurs in prokaryotes versus eukaryotes. Because in prokaryotes, the DNA can be read and produce RNA, which then can immediately get turned into a protein because it's all floating here together, right? It's kind of cool. Good. Prokaryotic ribosomes are a little different than eukaryotic ribosomes. We're not going to worry about any details about that, but they both have ribosomes. That's the key thing. It's totally going to be on your midterm, all right, that prokaryotes and eukaryotes both have ribosomes. It's absolutely going to be there. Eukaryotes. The DNA is in a nucleus that is bound by a double membrane. So the nuclear envelope, we call it, which is the thing surrounding all the genetic material, the nucleus, right? It is made up of two membranes. So there's two layers of membrane, right? And remember, all of these membranes, I've given you the caveat here, right? That archaeans don't all use phospholipid bilayers, okay? So I'm going to now speak in generalizations. All of these membranes are made up of phospholipid bilayers, hydrophilic heads facing out into the fluids with hydrophobic tails, all right, in between, creating a hydrophobic domain. And that is a phospholipid bilayer. That's what all of these membranes are made out of, right? Is phospholipid with its tails, phospholipid with its tails, making a big band of hydrophobicity that prevents everything from moving around freely. Now, when we say that the nucleus is bound by a double membrane, that means it's got a phospholipid bilayer and then another phospholipid bilayer, okay? So that means it's phospholipid, 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 phospholipid. It can get really confusing because bilayer, right, and then double membrane, they're both talking about two, okay? And they're very different. So every individual membrane is a phospholipid bilayer, right, which is made up of phospholipids associated because of their hydrophobic tails and their hydrophilic heads. But a double membrane has 2 layers, okay, of phospholipids. Now, if this was only going to relate to the nucleus, I wouldn't make such a big deal out of it. But it's going to matter a lot when we start to talk about complicated organelles like mitochondria and chloroplasts, which have multiple layers of membrane, each of which is important for different functions. But don't worry, that's not for another lecture or 2, or maybe 3 even. Cytoplasm, the cell juice, this is in the region between the plasma membrane and the nucleus. I already sort of explained this, right? Every time there's a membrane, it means that the fluid inside and outside of the membrane is inherently different in some way in the cell. So if it's cytoplasm in the cell, and then there's another membrane floating in the cytoplasm, the fluid inside of that membrane must be different. Cool? We need to give it another name, and we will. Awesome. I got 1 thumbs up. I'm calling that a clear to keep on moving. Eukaryotic cells are generally much larger than prokaryotic cells. Generalization is not always accurate. Question?
Is the phospholipid bilayer the reason why DNA and RNA processes can happen at the same time? Or is that more the cytoplasm? Like, do they have to go through that bilayer?
So, yes, and I want to clarify the language. All right? The fact that there's no boundary around the nuclear material, around the DNA, all right, means that you can go DNA, RNA, protein, boom, boom, boom. The presence of the nuclear envelope means that you have to go DNA to RNA inside of the nucleus, right? Because that's where the DNA is. Then the RNA needs to be exported. It does have to cross the double membrane here, the thing that we call the nuclear envelope. It has to cross the nuclear envelope to actually reach the ribosomes, which is exactly what you just said. I just added more words to it to make it more complicated. Cool? Was that a successful answer? Awesome. All right. Any other questions so far? All right. What did I say? Now I'm forgetting the 3 things that I said were the most important things in biology. Surface area to volume ratios, osmosis, and something else. Hydrophobic and hydrophilic. Thank you. Points to both of you. Very, very good. Hydrophobicity and hydrophilicity, right? It's going to continue being hugely important. Keep an eye out for it. It's not just that I'm obsessed and I'm bringing it up. It's what your textbook talks about all the time. You're going to see it in your labs. You're like, wait, is this only working because this is hydrophobic and this is hydrophilic? And I'll be like, yeah. Isn't that ridiculous? All right. So here's surface area to volume ratios. Oftentimes in a textbook, it'll just say the surface area to volume ratio is the limiting factor for cell size. All right. Cells can reach a maximum diameter because of their surface area to volume ratio. And it's a little more complicated than that. If the construction gets too noisy, I'll just get louder. This won't get annoying, right? If I'm just yelling at all of you, it's just to get over the sound. I'm not upset with you, I promise. All right? Surface area to volume ratios. Check this out. It's just math. What would you call this? Geometry? Right? The only thing I know about that field of study is it's what you say when a witch turns you into a tree. Gee, I'm a tree. Thank you. Yes. No, I know. I appreciate it. Okay? So here's basic geometry. The equation for the surface area of a sphere is 4 times pi r squared. You don't have to memorize that for this class. The equation for the volume of a sphere is 4 over 3 times pi r squared, r cubed, right? I could run example math through Excel. I've done that before in semesters, but I don't think it's actually really necessary. Most people are willing to trust me that as you increase the radius of something, of a sphere at least, as you increase its size, the volume will increase, it's cubed here, the volume will increase more rapidly than the surface area. So as you blow up a bubble, okay, there is more volume relative to surface area, the larger it gets, okay? As you blow up a balloon, think about it, right? As the balloon's really small, there's a lot of surface area, there's very little volume inside of it. But as you blow the balloon up, there's less surface area relative to, bless you, the volume inside of the balloon. Question? Absolutely. So metabolic requirements relate to, well, metabolism in general relates to all the chemical reactions going on in a cell. So if we talked about our basic chemical reaction of reactants and products, right? Metabolic requirements means you need all of the necessary reactants and you need to get rid of like byproducts, right? So when you've got a big old cell like this, it can accumulate waste inside and become depleted in essential resources. Whereas in a smaller cell like this, it can export waste or unnecessary byproducts across the membrane, and it can acquire new resources. The key thing here is that to get more stuff into the cell or to get stuff out of the cell, it has to go across the cell membrane. So you need enough surface area to service the volume. If there's not enough surface area, you can't meet the metabolic requirements of the cell. Solid, great question. Awesome. So, not every cell is a sphere, okay, but most like single-celled organisms are close to a sphere, and so they follow the same basic rule. The level of math that I ever needed to get up to be a biologist, you spend a lot of time just taking complicated shapes and breaking them down into spheres and cubes and triangles and well, cones and stuff anyway, because you can, those have simpler equations. So this is a generalization for roughly spherical cells. But I already told you that there are some cells that are very big, like a nerve cell, right? That goes from your head down to, this only reaches my knee. But a nerve cell going down to my knee. I got cells in my body that are this long. It seems that would break the rule, but it doesn't, because the surface area to volume ratio along the length of the, let's just call it a tube, all right? The cylinder's better. That's more accurate than a tube because it's not hollow. Still meets the rule, right? Still follows the rule. Stuff can get in fast enough, stuff can get out fast enough. Make sense? Cool. But surface area to volume ratio is hugely important in determining the upper limit of cell sizes. Okay, so how big can a cell get? Well, partially depends on the surface area to volume ratio. Cool. Awesome. Here's a eukaryotic cell. It's a hypothetical eukaryotic cell. It's a hypothetical animal cell, all right, in that it doesn't have any chloroplasts, or I could be more accurate and say it's a non-photosynthetic hypothetical eukaryotic cell. Not every eukaryotic cell will have all of these structures, okay? For example, not every cell on your body has flagellae, the plural of flagellum, all right? That'd be really creepy if all of your skin cells had a little projecting flagellum that like whipped around. It'd be eerie. And then that's how you swim. Could you imagine you just get in the water like this and then those whip and that moves you through the water? It'd be out there. All right. And not every cell has microvilli on it. You ever play Earthworm Jim 2 on like Sega or Super Nintendo? Yeah, I know. Don't worry. If you have a Switch, it's on the collection, that you pay monthly for. Play Earthworm Jim 2. You play a level where you go through the micro village, all right? Where you're surrounded by these micro villi. You're going through some guts, okay? These micro villi are projections of the cell membrane. And look at them, right? You know what these do? They increase surface area. What? So like your guts want to have a lot of contact with the junk that you just churned up, right? These are technical terms. You want to have a lot of contact so you can suck out all the good juice, okay? Right? Microvilli increase surface area of a cell. So a cell will often, or a membrane will often fold up like this when it needs to increase surface area. And while I'll never test you on the phrase microvilli, the idea of a folded membrane to increase surface area, that's going to come up a lot, especially in mitochondria. You can already see a folded inner membrane here, and chloroplasts, okay? Solid. Let's look at the eukaryotic cell more. We've talked about how not everything will have a flagellum. Flagellum is the thing on a sperm that whips and moves things around. Okay, cool. Microvilli, they're projections to increase surface area, increase absorption. Okay? Cilia are another projection that allows single-celled things to move. They're not even indicated here. But there are certain things that are going to be common to every eukaryotic cell. Things like, and I shouldn't say every, almost all eukaryotic cells, okay? Things like the endoplasmic reticulum, all right? The Golgi apparatus, lysosomes, mitochondria, cytoskeletal elements, including microfilaments, intermediate filaments, and microtubules. A centrosome, which is essentially an organizing center for creating cytoskeletal elements. We don't talk about it very much. Peroxisomes, which are essentially little things that can break things down, all right, like lysosomes. They get their name because a byproduct of their reactions is to produce hydrogen peroxide. They're involved in decreasing the amount of reactive oxygen species in your body, if you've heard of reactive oxygen species and antioxidants and things like that. Peroxisomes, they do matter, they are important, and we don't talk about them. All right? Trust me, if you're curious why, just look them up and try going down the rabbit hole to find out what they actually do, and you just end up hitting this wall of like, they have many functions, which is the most infuriating thing when you're reading primary literature articles. And then you find their source they cited, and that's what their source that they cited said, was that they had many functions. And then you look up Campbell biology, and they're like, they have many functions. It's like, I gave up. I tried year after year to make them interesting. They were never interesting. All right. Nucleus, DNA, mitochondria, endoplasmic reticulum, chloroplast, Golgi apparatus, vesicles, cell membrane. Key stuff inside of a eukaryotic cell. Okay? Let's start with these components in the cell as a factory analogy and go from there. Because there's only one other, maybe two other things that you really need to know about in the eukaryotic cells that aren't on this diagram. Casey Liu was a professor here who retired happily and now travels with his family. He's a great person from what I could tell. And he used to have this exact picture outside of his door. So I'm keeping his legacy alive in using this exact picture for the cell as a factory analogy. Also, he originated Lou points, he'd call them, and give them out to people. And now I give out John points, and everybody loves them. All right, cool. So I hope he's still doing great. Let's put some positive vibes out there for Casey. He lives up on Fickle Hill, so shoot him towards Fickle Hill. I hope he picks them up. All right. I don't know about you, I had a really good weekend. I'm feeling like nice today. But, all right. Cell is a factory. You excited? Oh, wow. Wow. All right. So, here we go. In a factory, you've got the headquarters. Within the headquarters, and this area is blocked off from all the lesser workers, because you wouldn't want one of them to accidentally spill their coffee on the blueprints, would you? So you keep them in a safe little room surrounded by a nuclear envelope. In the nucleus, the control center, you've got the blueprints for how to make everything that the factory makes. All right? Very cool. To make the things that the blueprints encode for, well, you are going to need power, okay? You need power generation, which you get from the mitochondria, okay? Very good. Then you need a separate site that actually produces the product, all right? In this case, it's proteins. These are just boxes that say proteins on them, all right? And you know that the protein-producing machinery, the ribosomes, right, are the key component here. But those ribosomes, all right, they may be embedded within the endoplasmic reticulum. So the endoplasmic reticulum, it's the majority of the membrane inside of a cell. It's just a big sack of membrane that gets all like bundled up. And it has all these ribosomes studded up in it, okay? And it makes other things as well. But it's really convenient when you've got ribosomes and membrane together. Because then you can sort of like just take that protein you make and go bloop and put it in a little sack of membrane. All right, a little phospholipid bilayer. And now it's safe. So you take the thing that you made and you put it in a little box. You take that box to another sack of membrane, and then that membrane sac opens the box, polishes it up, connects a few pieces, puts it in a different box, and that area is the Golgi apparatus right here. Okay, cool? In order to get food for all of the functions that are going on inside of the cell, all right, you can have a kitchen inside of the factory, or you can send out for food. This is the division between between a autotroph, meaning self-feeder or self-eater, and a heterotroph, meaning other eater or other feeder, okay? In an autotrophic organism, you make your own sugars right there, all right? And then you can feed those into the mitochondria, break them down, boom, 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 get the energy you need to make your proteins. If you're a heterotroph, you have to eat something else. You have to DoorDash it, all right, by consuming like a gazelle or something like that. Okay, cool. So, food production can be the chloroplast. The sorting can be the Golgi apparatus, all right? And then the packaging and transportation can be vesicles. What's a vesicle? It's a little bubble of phospholipid bilayer with some stuff in it. Cool? Cool. We're also going to talk about vacuoles, which are larger bubbles of phospholipid bilayer with some stuff in them. These are very general terms. So vesicles are just little sacs that then can be shipped around throughout the cell, even exported outside of the cell. Okay? And then you've got the walls. The factory must have walls. And so we call that's the cell membrane right there. And in an autotroph, you make the high energy sugar and then you burn the high energy sugar. You make the high energy organic molecules and then you burn through the high energy organic molecules that you made. So it makes you an autotroph. All right? How does a cell as a factory sort of work for folks here? Now we'll run through a video. I guess I'll play it. I hate these videos. A tour of an animal cell, okay? Since we started a little late, let's watch the video, then do our 5-minute break, and then come back and wrap up the lecture. Sound okay? It's like a 4-minute video, I think. Hopefully the microphone for the recording picks this up. I'll never update these. The only thing that I like about them is that they're so, is that they are corny. Let's make sure our audio is turned up. Woo! How's that? Let's try it again. Let's do it. Here we go. Ready, everybody?
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Pretty good, right? Whoa, look at this place. All right. So one thing I want to point out from that video, while it's still fresh in your mind, is you saw how vesicles were budding off of things and then fusing onto other membranes, right? That's because it's all phospholipid bilayer. And because it's all the same fundamental material, that hydrophobic stuff, the hydrophobic domain, they can fuse and break off of one another. And that's going to be hugely important for subcellular function. Just the fact that the entire membrane system is built up of phospholipid bilayer. All right? So 5-minute break. Let's go ahead and start it right now and begin. All right. I'm going to pause the recording. I'm going to stop the recording. I'm going to pause it, see if it works. It didn't. It should seamlessly stitch them together, saving me an editing step, which would be awesome. Did you know that Adobe products freaking suck and they're terrible to use? Yeah. I'm trying to use Premiere just to reduce the buzz in the background of these and then stitch the clips together and maybe put in like a fading transition if I'm talking to somebody. And it's been taking forever. It's terrible. Just work. Just use the resources more efficiently. I don't know. That's what we're going to talk about the rest of today, is the problems with Adobe Creative Cloud. But did you know that as a student at Cal Poly Humboldt, you actually have, included in your tuition, access to the entire Adobe Creative Cloud? So if you want to get into doing some Photoshop work, or do some more advanced video editing stuff, you have access. If you're curious, come talk to me. I'll show you where to find the download instructions. All right? You actually have some cool software that you get free access to just by working here. It's great. Working here. You're working, Enough. So everybody have a good break? That's fine. It's too long. I know. Am I bleeding? Why am I bleeding? I have blood on my mug. This is no good. Okay. Whatever, it happens. I'm old, my skin's weaker. You know what I mean? So we were talking about animal cells, weren't we? Why don't we talk about plant cells? Because they matter too, okay? Big difference between plants and animals is the capacity for photosynthesis. While not all plants are photosynthetic, at the level of a Biology 105 course, all plants are photosynthetic, okay? Right? We're not going to talk about plants that don't do photosynthesis. Anyways, one of the most important things, okay, the capacity for photosynthesis. Here it is. It's carried out in additional organelles called the chloroplasts. There they are. So what are the big differences we see between an animal cell and a plant cell on this very generalized breakdown? I already pointed out that these chloroplasts exist right here. But can anybody else point out something that's different? Yeah. Cell wall. Super huge, right? For bonus points, what's that cell wall made out of? Oh. Not quite, but it starts with the same letter. No, the cell membrane's actually inside of the cell wall. What about this? Which class of biological macromolecules is the cell wall made out of? Carbohydrate, remember? Points, of course. It's a structural carbohydrate. That's the animal one. It's cellulose. Cellulose. Remember, polysaccharides, OSE, really standard, cellulose. And remember, that's incredibly important because it's got a lot of energy in it. We don't have enzymes to break it down, so we're chewing on grass all day, eating as much as we want, and it just passes through us as insoluble fiber. A lot of insoluble fiber is just cellulose, all right? When you take like, what are those husks? It comes in a big jug and you're like, blank husk, and you put it into your smoothie to give you more fiber. The heck's it called? Huh? Psyllium husk. Thank you very much. Yes, yes. That's all fiber is usually, is cellulose. You can't break it down. Your body passes it through. It makes it make more liquid in your intestines to move the things through. It pushes other stuff through your body. There's fiber for you in a nutshell. Okay. Or at least insoluble fiber. Question, please.
Is fiber something else the reason cell walls are so...
Like rigid like that? Yep. So because they're made up of cellulose, this structural carbohydrate, which has a good degree of structure, it's actually laid down in these, it's like a knitted basket. It's really like a woven basket in that it's got layer after layer after layer after layer of cellulose laid down and then weaving between layer after layer after layer of cellulose, like something that you wove on a loom. Pretty cool. It's pretty cool stuff. Good. So the cell wall, it's a barrier, right? Oh, and hey, also, if you've ever had to count carbs for a diet, right? You know how, let's say you're doing keto or something, they'll tell you, look at the nutrition facts, take the carbs, and then subtract the fiber. Well, why do you have to do that? Because the fiber is mainly cellulose, right? Your body can't break it down, but it's still a carbohydrate. So that's why nutrition facts will have carbohydrates and then fiber. And if you're doing a diet that's low carb, you don't include the fiber. Because while it is a carbohydrate and included in the carbohydrate quantity, it's not one you can break down. Isn't that interesting? A little bit? I don't know. It's interesting to me, and that's what matters. Moving forward. The cell wall, it's a big, important barrier, okay? A plant cell is encased by this sort of box. That allows it to develop a bunch of internal pressure and not explode, and also can give a tissue, a plant tissue, structure. Remember when I blew up the glove like it was a leaf? I made internal pressure, and then it got structure just because it was pressing out against a boundary. That's what this cell wall, one of the things the cell wall does. It's also a problem, though, because if you live in a box, okay, how are you going to get rid of your waste and get the food things, get the things that you need? How are you going to meet your metabolic requirements, right? How are you going to do that? Well, you got it? Plasmodesmata, all right? So these plasmodesmata are basically little holes in the box. If there's ever a terrible situation where I'm living in a box and you're in charge of that box, please put holes in them, in that box, and please call them plasmodesmata. And remember, yeah, I would really appreciate that. All right? So these are sort of just channels that allow material to be exchanged from cell to cell. So you don't run out of stuff within the cell or generate too much waste and have no way to get rid of it. All right, what's another thing here? We kind of got the big one, the cell wall. I stole chloroplast. That was kind of cruel of me. But what's the other thing here that we didn't see in the animal cell? Central vacuole. That's exactly right. You can each have half a point. All right. Central vacuole. There it is right there. It's a big sac. All right. Everything inside the cell is just a sac of membrane. Okay. With different stuff inside of it and different stuff embedded in the phospholipid bilayer that makes up the boundary of the sac. The central vacuole in a plant cell, it serves many purposes, okay? Some of those purposes are sequestering ions. You keep ions in that sac, and we'll talk about why you need these ions later. Also, this can provide a lot of turgor pressure because you can just pump this full of water and it'll push everything else out to the side and keep the cell plumped up, right? And turgid, which is so important in plant cells. In plant cells, a healthy plant cell is turgid, whereas a healthy animal cell is not turgid, because if it had internal turgor pressure, it would burst. Okay? Too much internal turgor pressure. Neither cell wants to be flaccid, which is the opposite of turgid. Okay? And if a plant cell loses turgor pressure, well, the cell wall stays up, but everything inside shrivels in. Okay? Because the plasma membrane is still holding everything together. It just all goes and like squishes down. Does anybody know the word for that? There's a word for when a plant cell loses turgor pressure and all the stuff gets sucked in and concentrated. There's no like expectation here. Huh? It is desiccated. It is dried, yeah, but maybe not completely dry. There can still be water present. It's called a plasmolyzed cell. The cell gets plasmolyzed. I bring it up now because of my obsession with repetition, and because in one of our later labs, when we're talking about osmosis, you'll need to understand the phrase of plasmolyzed cells. Okay? So I'm introducing it right now. Animal cell can either pop open with too much internal pressure, be healthy, where it's not a lot of turgor pressure, or be flaccid, where it's just like a sac, like an empty sac, all right, empty grocery bag. A plant cell can be turgid and healthy, or it can get plasmolyzed, where it loses turgor pressure and then it loses all of its structure. The plasma membrane gets drawn in. It's bad news for the plant. That's when a plant wilts, right? Is when the cells start to become plasmolyzed. Because the cell membrane box, it's kind of like a leather box, not really like a wooden box. Does that kind of make sense? Imagine a water balloon inside of a box made of leather. And that's kind of a plant cell. Does that work? Cool. This is why there's only 60 slides in this PowerPoint for week 2, is because we spend a lot of time on each slide. I really don't want to do it. We want to watch let's watch the video. They're short, but they feel like a million years. What's this one? 1 minute and 45. Okay, ready? Everybody ready? Let's get laser focused because they're going to pack so much interesting material. Get excited. Here we go.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
The next time you pass by a plant, remember that we have more in common than meets the eye. When I was a child, I won an essay competition for Arbor Day, and I ended it with, "Now if you'll excuse me, I have to go water my best friend." And that's almost as bad as what they did there. You know? Horrible, horrible stuff. Anybody know what we call this structure that we're zooming into the plant through? It's an S. Stomata. These are the stomata. So we'll talk about these a little bit, but they're more botany talk. But they're little like holes that open and close like this, using turgor pressure actually, where the cells go brrr, swell up, or then they release water. Yeah. Anyway, that wasn't so bad. That wasn't so bad. We have more in common than meets the eye. All right, cool. Except we're cool and have TV, and they're dumb and sit in the dirt all day. So take that, plants. All right, there's our plant versus our animal cells. As you saw in that video, they focused on the cell wall, the chloroplasts, and the central vacuole. Exactly the same things that I tried to focus on in our discussion as the differences between them. Remember, still got to have mitochondria, right? Still got to have ribosomes for sure. Endoplasmic reticulum, Golgi apparatus, all going to be there. And both those videos discussed the cytoskeleton, or at least referenced it, and we are going to talk about the cytoskeleton and what it does. Don't worry. Okay. You know already that I'm basically in love with the plasma membrane. Why don't I marry it? I would if I could. I'll have to settle for marrying a human who is mostly made up of plasma membranes. But sorry, taken. Plasma membrane. All right? You know how much I care about the plasma membrane. It's made up of phospholipids. It's a phospholipid bilayer, right? Hydrophobic tails, they associate, make a hydrophobic region. Hydrophilic heads, they face the extracellular and intracellular fluid, and they associate with it because it's an aqueous solution, right? If you drop a bunch of phospholipids into a solution, then they'll auto-assemble into a phospholipid bilayer. I'm just running through the greatest hits of what I've said about the phospholipids so far. For bonus points, who remembers the type of molecule that a phospholipid is? All right? It's a whole class of molecules. Like,
Make soap out of it.
you make soap? Yep. It starts with an A. There's some points. Amphipathic. I know, it'll drive you nuts. But don't feel bad if you're not remembering these words. I'm throwing, like, probably in each lecture, 30 to 60 new words at you. Or that I'm telling you, like, memorize all these. So no need to feel any shame if you forget one of these words. This is an amphipathic molecule, just means that it has a hydrophilic and a hydrophobic region, or in other words, that it has a polar and a non-polar region. It's all about polarity. Phospholipid bilayer. What is it? It is a selective barrier. What does that mean? It means certain things can cross it and certain things can't. You'll also hear these referred to as semi-permeable membranes, all right? And the language, the overlap between those terms can be frustrating. I prefer semi-permeable in that certain things can cross it. Because selective implies that it is selecting for certain things to cross. But here's the problem, right? The phospholipid bilayer itself can't really select what's going to cross it. But the plasma membrane, through all of these special transport proteins, okay, can select certain things and say, let that in. Like this channel protein here is probably designed to allow certain molecules to go in or out of the cell. All right? So it is a selective barrier if we're talking about the plasma membrane, because the plasma membrane, and I'm sorry to be so circuitous with this, the plasma membrane includes the phospholipid bilayer and all of these proteins embedded in it. Cool? Yes, please.
So are the proteins what lets certain things through while the phospholipid keeps them?
Yes, and the phospholipid bilayer itself does not keep everything out. Certain things can move across the phospholipid bilayer, and the phospholipid bilayer is permeable to different things at different rates. But it all comes down to the hydrophobic region here. This hydrophobic region is the barrier that prevents totally free movement across the plasma membrane. Okay? So molecules that are highly hydrophilic, like ions, they won't really cross unless there's, say, an ion channel that specifically allows ions to cross the membrane. All right? Don't worry, we're going to do a whole talk about what things can cross a phospholipid bilayer, what things can't cross a phospholipid bilayer, and what things can slowly cross a phospholipid bilayer. I'll tell you right now, the most frustrating molecule is going to be water, because you would think that if the hydrophobic region is what's preventing movement of molecules across the membrane, then water definitely won't cross, right? That's just intuitive. But water can cross a phospholipid bilayer, just not that rapidly, okay? But that's what osmosis kind of is water crossing membranes like this. Okay, cool. So, the plasma membrane, it is a selective barrier. Certain things can cross it, certain things can't cross it. It's got a phospholipid bilayer. It's got a bunch of proteins embedded in it. Some of these are what we call glycoproteins, proteins with carbohydrates attached to them. All right? Usually the protein is anchored in the membrane, whereas the carbohydrate side chains project out and are used for things like identification by the immune system, for example. These carbohydrate side chains are what determine blood type, okay? If your blood cells have A-type carbohydrate side chains, you have A-type blood. We'll also have a whole discussion about blood types. Should be good for everybody. Okay, the plasma membrane is also referred to as the cell membrane. Tons of overlap here. Here we go. Phospholipid bilayer with inserted molecules. All right? It is semi-permeable or selectively permeable. Certain things can cross it. The proteins can be selective for certain things crossing it. Let's look at a couple of other things that are embedded in the phospholipid bilayer real quick. Cholesterol. All right? That's the only one actually we need to really worry about. We've got some different types of proteins here, okay? And we also have cholesterol, okay? Now, I've talked to you about cholesterol as well. Does anybody remember the class of biological macromolecules that cholesterol fits into? So, fat, lipids, yep. Points all over the place, okay? Very good. Cholesterol is a steroid. It is a lipid. It is a bunch of carbon and hydrogen, okay? Meaning it's very nonpolar, meaning it's nice and soluble inside of that hydrophobic region of the phospholipid bilayer. Now, I talked to you about a key role cholesterol plays in cells, but it wasn't a highlight, so I'm just going to give it to you again. Having cholesterol inside of a phospholipid bilayer helps to buffer its fluidity, keep the membrane from falling apart. So imagine we've got the phospholipid bilayer, right? The cell membrane. It's roughly, you'll see a slide that says this, it's roughly the consistency of salad oil. So it's like an olive oil bubble. So you've got the phospholipid bilayer, and you do not want it to freeze and shatter. That's bad. If you ever put olive oil in the fridge and it freezes and you can crack it, you don't want your cell membranes to do that. So you don't want your membranes to solidify, and you don't want your membranes to get too warm and get too fluid and then flop apart, all right? Because then you break the barrier between the outside and the inside of the cell, and your cell's done, right? So cholesterol is very useful as a buffer, meaning it does not make the membrane more fluid, and it does not make the membrane less fluid. That's key. But it will prevent changes in fluidity of the membrane. Because if the membrane tries to solidify, well, you've got these cholesterols sort of blocking the molecules from associating together and making a crystal. Or making a solid, all right? And if the membrane starts to wiggle too much and the energy is going boo, 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 passing between the molecules, well, having a cholesterol molecule stuck in the middle means the wave is coming, and then it hits, and cholesterol sort of just goes like, and absorbs some of it, preventing the energy from being transmitted to all the other phospholipids. So it's like trying to do the wave, but like holding hands and trying to do the wave, and there's somebody in the middle holding hands who's just like that, like really not feeling it, okay? Yeah, it's okay. All right. Sick. So, why do you think I highlighted that so much? Because it's going to be on an exam or what, I'm concerned with you getting all the points you can possibly get. Cholesterol, the idea of buffers, I think that cholesterol as a fluidity buffer, having questions about it is good because it makes sure that you understand what a buffer is and how a buffer is different from something that, say, sets a value. So I'm going to keep going. I'm just going to keep talking because that's what you're paying me for, okay? Does anybody remember how we set the fluidity of a phospholipid bilayer? What determines whether a membrane is more or less fluid? I'm throwing so many points at you. I'm sorry, but they're getting all over the place. The people around you are getting points just by having proximity to you, because that is the correct answer. How many of the fatty acid chains are saturated versus unsaturated? Specifically, if you have more unsaturated cis fatty acids in these, there will be a kink in the chain, and then the molecule can't reach as high of densities, and it won't solidify as easily. So more cis unsaturated fatty acids in the phospholipids means more fluid. More kinks, harder for it to solidify, right? More saturated fatty acids or trans-unsaturated fatty acids in the phospholipids, okay? The membrane will solidify more easily, but it also won't melt as easily. So animals raised in different climates have different degree of saturation, right? And if an animal, say, migrates from very warm to very cold regions, well, it may shift the phospholipid balance in its membranes during its migration. So if it's going from a warm to a cold region, well, as it moves to that cold region, it's going to have more cis unsaturated fatty acid containing phospholipids. See what I'm saying? But here's the thing. You can't make the membrane more fluid or less fluid by just dumping cholesterol into it. You can prevent the fluidity of the membrane from changing as drastically from, say, day to day by putting more cholesterol in it. All right? But that's the key difference here between a buffer and what actually sets the value. How does that feel? That was a lot of stuff coming at you. Question, please.
Saturated fats is more fluidity or
Saturated fats is less fluidity. The saturated fats are like dowels, so they can be packed tighter. They more easily become a solid. So butter is a saturated fat, olive oil is an unsaturated fat, which is a good way to sort of memorize that. Cool? Awesome. Let's continue moving forward. We're back. We're looking at the cell membrane. All right. Passive and active transport. Important concept. Some things move across the cell membrane. They have to, right? You got to get the reactants you need. You got to get rid of some products. Sometimes one cell is specialized to make something that the rest of your body needs, like insulin, okay? And you got to get that stuff out, all right? So in order to move it out of the cell, it needs to be transported. It can either be passively transported, okay, where it just diffuses out, or it can be actively transported, where it's actually pumped into or out of the cell. More accurately, I should say that it is pumped across the membrane. So some things just freely diffuse across the membrane passively and don't require any energy input, whereas some molecules, okay, they need to be actively transported. And active transport, it's active, right? It requires energy. And that energy is in the form of ATP produced by the mitochondria, yes. The cell does not really, it is a physical effect of the phospholipid bilayer that certain things need to be actively transported, and it's a rule of concentration gradients, right? Passive transport or diffusion, things always move towards equilibrium. If there's a lot on this side of the membrane and a little on this side of the membrane, all passive transport can do, if the membrane's right here, right? All passive transport can do is get those levels the same. But sometimes you want to concentrate something. So if I want to concentrate things on this side of the membrane's right here, I have to actively transport the molecules. I have to use energy to set up a concentration gradient. Because, check this out, right? Any concentration gradient is a representation, is order, is some degree of order. Order holds energy. Order is the opposite of entropy. Combating entropy requires energy. There are so many ways to present this concept. So active transport, right, is necessary when you're moving things against concentration gradients, okay? Otherwise, you can usually passively transport something if you don't need to really concentrate it by just making a channel protein. But the way the cell determines what gets transported is through what proteins it has in its membrane, right? And every cell, because every cell has the same genetic blueprints, every cell has the capacity to make every membrane protein. Regulation of which membrane proteins get expressed is one of the primary contributing factors to what makes each cell type different. They've all got a phospholipid bilayer, but two cells with different transport proteins will do very, very different things. All right? A lot more long-winded than maybe you were hoping for of an answer, but it gets to a really good point there. Okay? Cool. Cool. Active transport moves against concentration gradients using ATP. How did my example work there for concentration gradients? It was good? All right, they're gonna come up again, and then again, and then again, and then again. And then probably again. Sometimes you gotta get something real big into a cell, and it's too big to move through a channel protein. How do you do it? Well, you form a little vesicle. This is always hard. I know I can do this with my hands, but I never can do it quite right. Okay, there's a membrane around me. My brain, my head is the nucleus. Okay, my head is the nuclear envelope. My brain's the genetic material. Why not? Okay, cool. Here's the cell membrane. All right. Well, I want to get something in me, out of me. So I just go bloop. I push it against, and I pinch this off, and it makes a vesicle. And then that vesicle pinches off of the membrane and floats away and goes somewhere, okay? That's exocytosis, and it's necessary when you're dealing with very, very large molecules, or you're trying to move things en masse, right? You move a bunch of stuff. Also, let's say I want to get something in, right? Let's say that I'm a single-celled organism that eats things by bringing them into my cytoplasm and then dissolving them. That's phagocytosis, it's called. Okay? Well, in that case, I do the opposite, where I form the membrane in, okay, and capture something that was outside, and then I bring the little vesicle into the cytoplasm. Kind of make sense? That's endo versus exocytosis. You form vacuoles, which are just big vesicles, which are just sacs of beta-phospholipid bilayers. Anyway, so what's this whole slide about? This slide explains to you that the membrane separates the interior and the exterior of the cell, but certain things need to get in and certain things need to go out. Those things can move in and out either via passive transport or diffusion, where concentrations equalize. Sometimes you want to pump something across and concentrate it. That requires active transport and it requires energy. And for very large molecules or very large amounts of stuff, you need to take the membrane and make vacuoles out of it and then move the vacuoles around. Question, please. What's that? It doesn't break down the membrane. It's more that the membrane, so it went from being my cell membrane to a vacuole, and then it goes, let's say that it goes to the Golgi apparatus, right? Then it goes to the Golgi apparatus and fuses there. And it's still part of this whole membrane system. And then the Golgi apparatus, or the endoplasmic reticulum, breaks a little piece of membrane off and replaces the membrane. So inherently, it reduces the overall surface area because you pinched off a piece of the membrane. But then it will be replenished. Was that what you were getting at? Sick. That's very good. So yes, just inherently, it does reduce the amount of membrane on the cell membrane, but your body has the ER to keep making more and more membrane. Yeah, awesome. Okay, we're just going to push all the way to our 12:05 ending time. Because, there's a lot of slides to get through. Nucleus. All right. The nucleus. It is a double membrane bound structure, meaning it has two phospholipid bilayers, right? The nucleus contains most of the cell's genes or DNA. It says most here because there is DNA inside of mitochondria and chloroplasts. They have their own genome. We call the DNA there extranuclear DNA because it's not in the nucleus, okay? But most of the DNA in a cell is in the nucleus, okay? So this is the nuclear genome is also how it's referred to. There's also a term for it, In a eukaryote, this is all eukaryotic stuff, by the way, right? Because there's no, are we comfortable with that? In the prokaryotes, it's all super simple. Cool. It's all eukaryotic stuff. In eukaryotes, the DNA is broken into these little discrete units Each discrete unit is called a chromosome, okay? Chromosomes get compacted down. The DNA gets wrapped around these proteins, okay? The DNA and protein complex is called chromatin, all right? The language starts to get really frustrating when we get into genetics. We'll talk about what a chromatid is, which is different from chromatin. It's very annoying, okay? But you have these discrete units called chromosomes. Chromatin is the DNA protein complex right here. It's important for packaging. There's a region called the nucleolus in here, okay, which is where ribosomal RNA is made. I've told you before how there are different types of RNA, and not all RNA ends up being translated into a protein. The ribosomes, those little non-membrane bound organelles, they actually have RNA in them. They are part protein and part RNA, all right? And the RNA in a ribosome is named ribosomal RNA. Nothing too wild there. The nucleolus is this little special region where you just make ribosomal RNA. I won't be testing you on that term though, right there, but it's interesting. So here's the last thing I'll say today. Look at the nucleus, look at the nuclear envelope, and notice how it's actually continuous with the endoplasmic reticulum, okay? Remember how I told you ribosomes can be embedded in the endoplasmic reticulum? Well, if I have to go through all this hassle of taking RNA, exporting it out of a nuclear pore, and then hoping that it finds a ribosome, how about just pumping it directly into a continuous chamber of the endoplasmic reticulum? So anyway, the outer layer of the nuclear envelope is continuous with the endoplasmic reticulum. Okay, let's see what our next slide is, where we'll start. Oh, nuclear envelope pictures. Cool. Ribosomes, sick. So let us keep moving forward. Tomorrow in lecture on the cell. How far did we get? I'm just curious. We are on slide 17 of 80. That's perfect in a 4-day, week. So we'll hopefully get, we might get ahead of schedule. Who knows? Anyways, we'll start our lab at 1. Please print a protocol and bring it to lab. If you haven't started assembling a lab notebook yet, it's not too late. 3-ring binder. I got a 3-hole punch here. If you have any questions, feel free to come talk to me. And that's the end of our lecture for today. Thank you, everybody.